Here we present a robust protocol for the detection of bacteria within biopsied bladder tissue, which has broad applications to many tissue types where bacteria are suspected to be present. The main advantage of this technique is that it's unbiased. It allows the researcher to detect bacteria within tissue without any a priori knowledge of which bacterial species is present.
This technique may help determine if bacteria have invaded the bladder walls of women with recurrent urinary tract infection and inform therapeutic choices such as the selection of tissue-penetrant antibiotics. While designed for bladder tissues, this technique can be broadly applied to other tissues with resident bacterial populations with minimal optimization. Individuals who are new to this technique may struggle with cover slip placement and imaging. Focus on optimizing cover slip placement to reduce bubbles and practice imaging on non-precious samples.
Helping to demonstrate this procedure will be Jashkaran Gadhvi, a graduate student in my lab.
To begin this procedure, clean an empty fume hood or appropriately fitted biosafety cabinet with 70% ethanol. Prepare all of the necessary buffers and reagents as outlined in the text protocol. Next, clean 5 Copland jars with 70% ethanol and let the jars dry. When the jars are dry, label them as shown here and fill them with 100 milliliters of the appropriate solution. In the fume hood, place 2 sides per biopsy into a vertical slide rack.